Bio-Rad has produced a new cutting-edge QC performance assessment and design technology. This video uses simulations of a glucose assay to illustrate and explain some of the key concepts that are involved in our computations. Let's begin by orientating ourselves. Glucose results are represented on the vertical axis, and time is represented on the horizontal axis. Let's assume that our glucose method has a coefficient of variation of 2.5%, and that we've established an allowable total error criterion, our quality requirement, of plus or minus 10%. Now let's imagine that we are in the laboratory doing routine testing. We expect results to be evaluated over time. Here we can see the distribution of results. In this case, they range from about 50 to 300. We recognize that the results we are recording are not perfect, and that each result has some degree of uncertainty associated with it. In reality, each of the dots would be better depicted by a vertical line that represents uncertainty in the measurement result. In this particular case, where we are assuming a CV of 2.5%, large results like the glucoses that are around 250 have more uncertainty associated with them than the smaller results that are closer to 50. Here we see the simulated measurement errors for the results displayed above. These were randomly generated, assuming a coefficient of variation of 2.5%. The lower right panel is a histogram of the measurement errors. Now let's continue to measure results. Again, here we see the results as well as the distribution. Note the horizontal dashed lines at plus or minus 10. These are the limits set by the quality requirement. Likewise, the vertical lines in the right panel serve the same purpose. We can compute and predict the likelihood of producing a result that fails to meet the quality requirement by the number of observed measurement errors that exceed plus or minus 10%. If you look at the lower right panel, there are no results that exceed the limits. This is because we have a TEA of plus or minus 10%, but a CV of 2.5%. So the chance of getting a result with a measurement error as high as 10% is very low. In fact, in this case, percent question mark representing the percent unreliable is 0 when rounded to the nearest 1/10%. Let's look at a different case. Before, we used the CLIA recommendation for our TEA, 10%. Now let's use 6.9%, a specification used in Belgium. We'll only change that parameter and then rerun the simulation. Notice that the distribution of results in the upper and lower panels are unchanged. But the locations of the dashed lines have changed. They are no longer at plus or minus 10%. They are now at plus or minus 6.9%. Also notice that we now have a result with measurement error that exceeds 6.9%. It's denoted in red. It doesn't exceed 10%, so it wouldn't have been red earlier. Let's make another observation about the result that isn't meeting the quality requirement. There is nothing about this result that would give a laboratorian a clue that it contained an unacceptable amount of measurement error. If we continue to measure results, we see a few that are failing to meet the quality requirement. The histogram is the same as it was before, but now we see that 0.6% or 6 out of 1,000 fails to meet the quality requirement. Looking at data in this manner turns out to be an interesting way to appreciate the likelihood of producing results that fail to meet the quality requirements. However, in quality control, we're concerned with the ability to detect out-of-control conditions when they occur, before they produce too many bad results. So let's take a look at that case. The upper panel shows the same timeline for glucose results. The lower panel shows measurement errors again. But now we are going to include quality control results as part of the testing stream. We're going to test two levels of QC material every 50 patient samples. Level 1 is at 60 milligrams per deciliter, and Level 2 is at 130 milligrams per deciliter. The diamonds represent the QC results. We begin the process with a QC event. The measurements are around 60 and 130. Here we see the measurement error for those results. In this case, the measurement errors are both close to zero. We're going to continue to assume a TEA of plus or minus 10%, a CV of 2.5%, and that the lab is using a 1,3S, 2,2S, and R4S QC rule combination. These two QC results are within 1 SD of their assigned values. Therefore, the QC rule is accepted. So we're going to test patient results. The point in time where the yellow triangles appear is the point that a systematic 5% shift down in the process occurred. That happened 27 patient results prior to the next set of QCs. You can very clearly see the 2 CV shift downward in the measurement errors. Prior to the yellow triangles, the blue dots are centered around 0. Now they're scattering around minus 2S. It's easy to appreciate the effect of the 5% shift down in the distribution of measurement errors. However, if we look at the actual glucose results, it's not at all apparent that a shift has occurred. Here we see our next two QCs. One is between 2 and 3 SDs below its target, and the other is almost at its target, so the QC rule isn't going to be violated. Therefore, we continue to produce results that are adversely affected by this out-of-control condition. Now we see two results that fail to meet the quality requirement. Note that all of the results are being affected by the 5% shift, but only 2 of the 77 results that have been affected failed to meet the quality requirement of plus or minus 10% measurement error. If you look at the actual glucose levels for the 2 points that failed to meet the quality requirement, there is no indication that those points are not fit for their intended use. Let's continue testing. Here we get another unacceptable result. Notice that we are also keeping count of how many incorrect patient results have been produced. So far we've seen 3. 2 occurred prior to the last acceptable QC, and 1 has occurred since the last accepted QC. So, we've accepted QC 3 times since the 5% shift occurred, and we have 127 patient results that have been affected. So, we continue testing and get another acceptance and another unacceptable result. Now we have a total of 4. We continue testing and get another QC rule acceptance. Finally, on the 6th try, we get a QC rule rejection. We now have a QC result that is more than 3 SDs below its target. So in this case, where a 5% shift has occurred, it took 6 QC events to detect it. And 277 patient results were affected by the out-of-control condition. 7 of those 277 were unacceptable. 3 of those have occurred since the last acceptable QC event, and 4 were prior to the last acceptable QC. Let's say the laboratory experienced another 5% shift downward at a later date. Would we expect these exact same results? Would we expect it to affect 277 patient results, take 6 tries to detect, with 7 incorrect results? No, absolutely not. We would expect similar behavior, but not exactly the same because of the random nature of this process. So let's try this again. We have a QC event, and it is an accept. We run patients, and then the out-of-control condition occurs. This time it occurs 34 patient specimens prior to the next set of QCs. Let's continue testing. We have a bad result which we failed to detect. We continue testing, and at last we get a QC rule rejection. In this particular situation, it only took 3 QC events to detect the 5% shift downward. 134 patient samples were affected. 2 of the results were not fit for their intended use. 1 of those was prior to the last good QC event, and 1 was after. What if we repeatedly simulated this exact same out-of-control situation over and over? What we're seeing here are histograms of the quantities that we've been paying attention to. The upper left panel shows the number of unacceptable results that were produced prior to the last good QC event. We call this NUF. We can see that in half of the simulations there were no unacceptable results, but there was at least one situation where there were as many as 10. NUC represents the number of unacceptable results since the last good QC event. Here, in over 70% of the cases, we'd expect that number to be either 0 or 1. There were a few cases where it was as large as 4, but never greater than 4. NU represents the total number of unacceptable results. This is the sum of NUF and NUC. NQCE is the number of QC events to detect the out-of-control condition. In about 35% of the cases, that 5% shift was detected by the first scheduled QC after the out-of-control condition occurred. However, there was at least one situation where it took 10 QC events. NP is the number of patient results that were adversely affected by the out-of-control condition. % is the percent of the patient results that failed to meet the quality requirement while the out-of-control situation persisted. We see there is a wide range of possible outcomes that we could get from a 5% out-of-control shift. We can summarize our expectations by computing the averages for each of these distributions. If we do that, we find that the average number of unacceptable results prior to the last good QC event is 1.3. The average number of unacceptable results since the last good QC event is 0.9. The total is 2.2. The average number of QC events to detect the shift is 2.4. The average number of patients that were affected was almost 100. The percentage of the affected patients that were incorrect was 2.3%. Now let's look at a different magnitude of out-of-control condition. Let's look at a 7.5% shift up. This is a 3 CV shift up, which is really easy to see here. As we would expect, we have a greater number of results failing to meet the quality requirement. In this particular case, 3 unacceptable results were produced since the last acceptable QC event. With this large of a shift, we expect an increased likelihood that we'll detect the shift. It only took us 1 QC event to detect it. Would we expect to get the exact same performance if we did this again? Let's do it 100 times. We find that over 80% of the time, we wouldn't expect any QC results to be unacceptable prior to the last good QC event. This is due to the fact that in over 80% of the cases, we're detecting the out-of-control condition at the next scheduled QC after the out-of-control condition occurs. There are none that are prior. We see that the number of unacceptable results since the last good QC event can now be much larger. 0 in some cases, but as large as 10 in others. That's because the probability of an unacceptable result is greater with a 7.5% shift. If we look at the expectations for each of these things, the enough is 0.7, the ANUC is 4.5, so the total is 5.2. This is a larger number than we had before. The expected number of QC events to detect this out-of-control condition is only 1.2, so most of the time it would be detected on the first try. However, the percent of results that are incorrect when this out-of-control condition occurred is close to 16%. To understand the other end of the spectrum, let's look at a really small out-of-control condition. What if we had a 2.5% shift upward? This is a 1 CV shift up. The out-of-control condition occurs. We see that the points are shifted up and scattering around +1s, but that isn't a very large shift. Therefore, we don't see any results that fail to meet the quality goal. Let's continue to quickly run more tests. So far, we've had 336 patient results that have been affected by this one CV shift, but none of them have failed the 10% criteria. Note that we're on the 7th QC event and have not yet detected the out-of-control condition. We're now on the 14th try, and we finally see a result that has failed the quality requirement. However, we still have not detected the out-of-control condition. Finally, on the 21st QC event, we get a rule rejection. Over 1,000 patient results were affected by this 2.5% shift, but only one of them failed to meet the 10% quality requirement. If we were to simulate this out-of-control situation again, would we expect that it would take 21 tries? No. Let's test this. Let's simulate this condition 100 times. In this case, notice that the expected number of QC events to detect this 2.5% shift is quite large, over 15, but the total E-new is only 1. The reason is that we only expect 1 out of 1,000 results to fail to meet the quality requirement when a 2.5% shift occurs. So, we've looked at 3 different cases that are shown here. This is the average number of QC events to detect the out-of-control condition. This is the percent of the results that were unreliable during the existence of the out-of-control condition. This is the expected number of incorrect results that were produced prior to the last good QC event. This is the expected number of unacceptable results that were produced since the last good QC event. The circles show the expectations that we observed in each of those cases. If we wanted, we could continue to simulate different out-of-control conditions, starting at a negative 20% shift and going up to a positive 20% shift, in small steps of 0.1%. These circles would eventually create curves. Doing this many simulations would take a lot of time. Fortunately, we do not have to do that because we've developed the mathematical algorithms that can compute these expectations without simulation. We see that the mathematical algorithms compute values that fit the 3 cases that we actually looked at, and fit all those in between. These 4 figures that are produced are included on the summary page of our risk calculator. Visit www.qcnet.com for all of your QC needs.